something different every time I look at it. Okay. So here we are. Let me go back to, this is pain. There we go. So I entered all those ID numbers that I generated on that syllabus, the paper syllabus I handed out to you. So hopefully you wrote it down, but when we have our first test and we have our first lecture quiz this Friday, and it's covering chapter 1, it says it on the syllabus, please make sure and look at the chapter outline and the practice exam, okay? And definitely the flow diagrams, practice those too. That's what you're gonna need to prepare for that quiz. Now I do have some gaps. So if you forgot to turn that last page into me, please get it to me. Okay, so this is what we're covering today, chapter 2, part 2. It's going to be quick. We might finish it today. And then the handout for the practice exam. So lecture quiz 1, you're like, oh, where is all that stuff? Go back up to chapter 1 here. And then here's your study outline, the practice exam, the blank and the key flow diagrams. It's all up there with that chapter. Any questions about that? That's how you're going to prepare for this stuff. Yes. Chapter 1. Yep. If it says this is what it's on, that's what it's on. That's it. That's my contract with you. This is what I said it's going to be on. That's what it's going to be on. That's annoying. Okay. Very straightforward. Try and be transparent. I try not to leave you guessing. Okay, now what else? I think that was it. Okay, so let's start with a PowerPoint. Do I have it up? I think I do. Maybe not. Come on. Oh, there it is. Too many tabs. All right, so here we are, chapter 2, part 2, cells in the environment. Get rid of subtitles, those are a pain. God, just to get started takes forever. Okay, not next week, because I think next week we're doing cell metabolism, but the week after that, we're going to be doing cell transport lab. And so I'm going to cover the concepts that are going to be involved in that lab today. But believe it or not, everything I'm covering today, you had an intro bio, but I'm going to try and give you the physiology. I know this mask is muffling, the physiology and nursing application of those things, because I doubt they did it when you first learned it in 105. But we'll talk briefly about cell membrane permeability. And does it have application in nursing? It absolutely does, okay? So if a membrane is permeable, it's permeable, let me write it down, freely, come on, pen, with a better color. Nope. Freely allows substances across. You don't need channels, you don't need carriers, you don't need receptors, you don't need anything. Now, gases like oxygen and CO2 move this way across our membranes. If this curtain was a membrane, it'd be able to slide right on through the curtain with no problem, all right? But gas molecules are pretty small. Small things get across membranes very easy. Big things, not so much. So this is meant to show going freely across the membrane with no problem. Now, if you're looking at this PowerPoint PDF when you're online, the GIFs I've embedded in my PowerPoint won't be active, but if you wanted to see it, you click here and it brings you to the GIF where I found it. Now, our membranes are selectively permeable. There's a bouncer at the door. It's going to allow some to come in, but not others, all right? It's going to be more complicated. So selectively permeable is small substances pass easily, but large, and I'll put slash other substances, not so much. I'm not saying it's impossible for them to pass, but it's going to be more complicated. They're just not going to sail right on through the membrane. So I got the dog trying to get to kitties only because it's the concept, especially in lab. Small things can fit through the kitty door, but big things can't. That's exactly how our cells are. The dog could get through, but they would take a special receptor or something on the membrane. It's going to take ATP to cart that thing in. All right. Can happen, but it's hard. So here we've got small substances. This looks like it's water. And I don't know what this is. It doesn't matter. The big pink balls, they're obviously not getting through with this particular membrane. So that's how our cells operate. So this chapter, you can breathe a sigh of relief, is pure vocabulary. There's really no flow diagrams in this one. So it's all vocab. All right, so starting with types of transport. Passive transport. I know you had it intro bio. It's passive. Why is it passive? If anyone remembers from intro bio. No energy is required. Go back. Sometimes you touch something on the screen and there we go. Requires no ATP. Come on, pen. Here we go. No ATP. And here's another way of knowing it. Substances move with their concentration gradient. And that is always going to be for places where it's in high concentration, for places where it's in low concentration. They're running out of space. Okay, they go from high to low. That's why I have the person going downhill. Going downhill is coasting because you're going from a place of high to a place of low, and gravity to do the work for you. No ATP needed. That's the same thing with passive transport. All right. And so this is showing there's lots of these little red balls, whatever they are, on the left-hand side to start with, and not much on the right. So it's in high concentration on the left, low concentration on the right. Substances always want to go from where they're high to where they're low, and it takes no energy to do so. Now, the opposite of that is active transport, which, if it's opposite, it requires ATP. And I'll put requires ATP to move substances against their concentration gradient. And that would be from low to high. Okay, because substances, they don't want to flow that way. It's going to take energy. It's like salmon swimming upstream. It's going to take a lot of energy because you're going against the current. You're going against the concentration gradient. So there's a lot that happen to be inside the cell and not many outside. And this is basically a pump. And we'll talk about pumps. But they're a pump because they require active transport and ATP to make it happen. So if you're going uphill, you're fighting it. It's going to take energy. Okay, so we're going to start with passive transport because there's 5 types of it that I'm going to teach you. I'm going to teach you the relevance of it in nursing. So if you go to page 16 of the wiki text, let's try this real quick. No, we're not presenter view and show. Keep ink. Okay. I've got a lot of tabs. I got to do something about all these tabs. So it said page 16. So remember, if you go up to here and you type in 16, and I can't do it right now because it'll take you right to that page. So you don't have to scroll forever. All right. So I've made it very user friendly. And you can also just go to cell physiology, and that's page 14. And so I've got all my comments in there. So it's going to be very easy for you to navigate that. All right, back to this. Thinking. All right. Okay, simple diffusion. It's all passive, doesn't require ATP. Oh, I hate the red pen. Let's not do that. Equals substances move across membrane without the need of anything, without channels, without carriers. I'll put ETC. There's many different things that substances can use to get across a membrane. Simple diffusion goes right on through. So that really is oxygen, CO2, and some other things. All right, so nothing here on this membrane. Okay, I know it's a little busy, but that's the point. no ATP, no channels, carriers, whatever you need. So these substances move very easily. Now, diffusion with channels, they've got channels. Okay, so I think I don't even have to define it. It's simple diffusion with channels. But what I will say, diffusion with ion channels. We're going to be talking about ion channels a lot. And so I put here charged ions like sodium, got a plus sign, so it's charged, chloride, potassium, and calcium. Your cells are very careful about these charged ions coming across the cell because it can cause a cell to be stimulated or it can cause a cell to be inhibited. So I'll tell you a little something about our cells, and all of them are kind of arranged this way. So I'm on this side of a cell membrane. This is the membrane, right? On the other side of this curtain, the outside of the cell, is a bunch of sodium ions because cells keep sodium, a lot of sodium outside because sodium is always stimulatory. See, I put the S's, sodium is always stimulatory. If a cell were to allow all that sodium to flood into the cell is going to be depolarized. It's going to be stimulated, all right? So it's like and it will have an ion channel. So I think I have. So let's look at this real quick here. I know I wrote over it. Okay. This is basically an ion channel. It's a receptor, okay? And a signal from the cell here, which is going to be a neurotransmitter, when it binds, and I'll wait for it to cycle through. These things, yellow and orange, are a neurotransmitter, a chemical messenger that crossed the synapse. When that neurotransmitter binds to the ion channel, it allows an ion in. It's like opening the door. All right, so the neurotransmitter opens the channel, opens the door, and lets the ions in. And doesn't say what these are, does it? It might say it up here. I think it's sodium, okay? That's what this is. And so if you want to look at this GIF, you'd click here for it. But this is a very important one. And in chapter 4, which is the chapter that is closest to a nursing course that you're ever going to have in this course, it's going to be very important. These are going to be very important. You're going to need to know them cold. And then I'm going to start talking about sympathetic and parasympathetic responses. But we're going to start here. Simple diffusion requires an ion channel, and these are examples of ions. All right, so we'll come back to that later. So here, often involve a neurotransmitter that binds to the channel to open it or open the door. So that's how our cells work, but they're very careful. Sodium and calcium are always going to stimulate a cell. Potassium and chloride are always going to inhibit a cell. So the cell is very careful about what ions allows in because it has such a profound effect on the cell. Okay. Facilitated diffusion with a carrier. Okay. It is substances. It's very simple. I don't know why I'm putting it, but substances need a protein carrier. To cross the membrane. Okay. So just like this. All right. Now GIF stopped circulating and everything, but you can click down here. So look, glucose. We're doing a lab this week on glucose. Amino acids and some other things. Glucose is still a simple sugar. It's a monosaccharide. It's still pretty small, but it does need a carrier. It's not just going to sail right on through, and it doesn't get in with an ion channel either. So this is just another way of getting it into the cell. So the only reason this carrier would work is insulin. Now, I'm not going to get into the physiology of how insulin makes the protein carrier for glucose open up, but that's what happens. All right, and then glucose can enter the cells. So that's facilitated diffusion with carriers. So it involves carriers. Now, osmosis, this is an important one. Osmosis is the movement of water into cells based on tonicity of the solution surrounding the cell. Okay. So here's another way of thinking about it, because this is really how water moves. So over here, we have got water, the little blue circles, and then the green circle is sugar. So there's a lot of sugar on the right side of this membrane and no sugar on this side. What water wants to do is cross the membrane where the solute concentration of whatever is greater, and water wants to go dilute it. So that's another way of thinking about it, is water wants to move to the side, I'll say to side, of membrane with greater solute concentration. This is how I think of it. To dilute it. Water wants to dilute things. Okay, so if I put a lot of sugar outside, think of a cell sitting in a solution, like a petri dish. If there's a lot of sugar, glucose, or whatever in that solution, water is actually going to leave the cell, cross the membrane, and try and dilute that sugary solution in the petri dish. Okay, and the other way is also true. If the cell is in pure water, especially if it's distilled and everything, it's just water. There's no salts, there's no glucose, there's no proteins, there's nothing. Now, water in the petri dish has a low concentration of solutes. The inside of the cell has a higher concentration of solutes because it has proteins and sugars and things like glucose. Now, water from the petri dish will enter across the membrane of the cell and enter the cell. That's how osmosis goes. So, when you have to hydrate someone with isotonic, or also known physiological or normal saline, which is 0.9% NaCl salt, why can't you just have an IV bag of sterile water and put that right into their bloodstream? Why does that not work? Do you think it would kill the patient? Yes, it will. It'll kill the patient because of osmosis. Because what you're doing is you're introducing pure water right into the bloodstream with all your cells, red blood cells, white blood cells, platelets, and other things, okay? Pure water has a virtually no concentration of solutes. It's just water. Now, all the cells, your red blood cells, white blood cells, anything else, is going to be more concentrated inside. That water is going to cross the cell membrane and enter the cells, and the cells are going to go pop. Water is going to enter the cell is going to swell and possibly, most likely, rupture. And it can happen in about 30 minutes time. That's how long it would take to kill someone with an IV of pure water, because it has to do with tonicity. So you give them an IV solution with the same salt concentration that is found inside your cells. And if the concentration across this membrane is one salt concentration on this side of the membrane, like inside the cell, it's the same. Water won't go anywhere. It won't cross the membrane. There's no concentration gradient for it to try and dilute it. How many people know that's why you give isotonic saline? Now you do. If anyone ever asks you that, osmosis. That's it. That's the answer. Okay, so a hypertonic solution. This is the solution that's surrounding the cell. It has a higher solute concentration than inside the cell. So what's going to happen is water leaves the cell. That's what happens. So if I put a cell in a high fructose corn syrup solution, which is what we're going to do in the cell transport lab, that cell's going to shrink. It's losing water because the water is leaving the cell to try and dilute the hypertonic solution that surrounds it. So that's what will happen. Okay, the cell shrinks because it's losing water. And so that's what this picture is supposed to represent. You put it in a high salt concentration, a high sugar concentration, could be a high potassium concentration, could be anything. But water will leave the cell to dilute all those solutes that are surrounding it. And that can kill a cell too. All right. Now, the opposite is you put the cell in pure water. So the solute, you have a lower solute concentration outside the cell. Okay, water enters the cell. the cell swells and can rupture. Now, I might have gone over my image. I don't know. Apologize if I did. But that's what's going to happen. You put a cell like a red blood cell in pure water, it's going to swell up, it's going to rupture. And that would happen. It would happen very quickly. You're going to see in the lab, we're going to put a decalcified egg cell, which is a very big cell, and you're going to put it in a sucrose corn syrup solution, and the egg's going to shrink. When you take that cell and put it in pure water, within 30 minutes time, it swells back up again. That's fast. Osmosis works fast. Okay, isotonic solution, the solute concentration is the same outside as inside the cell. Is the same inside as the outside. Osmosis doesn't happen. And if osmosis doesn't happen, the cell stays the same. Okay, so normal or physiological saline is an isotonic solution because it's meant to protect your red blood cells primarily. Because if you don't give them the right concentration, you got problems. Okay. Everyone get a chance to write that down? By me writing it, it slows me down in the PowerPoint to give you a chance to write it as well. How are we doing on time? Pretty good. Now we have one more form, and here's the normal cell right here. Normal, happy red blood cell. So this is a GIF kind of showing it. You have got the cell, red blood cell, in pure water, let's say. It's a hypotonic solution. Inside of the cell is more concentrated. The water rushes into the cell, and that sucker's going to blow. If it's a hypertonic solution, so there's a lot of sugar, a lot of salt outside the cell, water leaves the cell to go dilute all those solutes, and the cell shrinks. A shrunken cell is also pretty much a dead cell. So to keep your cells nice and happy, you want isotonic, where everything is the same on either side of the membrane. Water will not move if there's no concentration gradient of solutes. It won't. You stop osmosis from happening. All right. Now, filtration is the last form of passive transport. And this is very important in your kidneys. So in anatomy, did you guys learn the kidneys down to the nephron stage? Some of you, yes, maybe some of you no. It's okay. It's really is physiology. So here's what happens in your bloodstream. So this is your bloodstream, all right? Inside an arterial capillary, there's going to be a lot of oxygen, glucose, things like that. Let's just think of the oxygen. Okay, at your kidneys, inside these things called the nephron, renal blood, kidney arterial blood comes to your nephrons, and there's millions of nephrons. And a nephron has an opening that's kind of like the head of a snake, and that arterial blood comes into it. Now, this nephron, the first thing is called a capsule. It has a semi-permeable membrane. Small things get across easily, big things do not. And what makes across these membrane pores are small things, gases, water, small sugars like glucose, they get through easily. Big things like red blood cells, white blood cells, all those things are too big. They'll never pass the membrane, so they get ejected out. What makes it across the membrane makes it across through fluid pressure, arterial blood pressure, right? Has pressure. That pressure is pushing those small things into the nephron. Once it's in the nephron, it's been filtered because it made it through the pores. That is the filtration that's happening. So filtration happens when arterial blood passes through small pores in the kidney nephron. I'll say nephrons because there's many of them. With the aid of fluid pressure. So in this, the blood is filtered. Small things get across the membrane, but not big things. But it's the fluid pressure that's pushing it across. So that's the filtration in that. Okay, so 5 forms of passive transport, all vocab. So the next is active transport. I don't think I have anything after this. I think it's active transport. Oh, this is, oh, I'm glad I had this in here. So this is arterial blood entering this capsule of a nephron. Small things make it across the pores in this membrane, and they're small pores. Only small things get through. Big things that are too big to get across the pores get ejected out of that nephron. And the stuff that makes it into the nephron, this is all filtrate. It's been filtered. It was pushed across a membrane that had small pores. So that's what it would look like. That's what's happening at your kidneys. Your kidneys are filtering all your arterial blood, about your entire blood volume, about 5-something liters, in the 40 minutes that you're sitting here. That's a lot of filtration going on, and that's the passive transport method by which it's happening. Okay, so active transport. I could talk a lot about it, but I'm not. I want to keep it relevant to what you need to know as future nurses. So it's the movement of ions with a pump fueled by ATP. Remember the ions that I said the body is careful about letting through: sodium, calcium, potassium, chloride. They can have a powerful impact on a cell if they're left to enter very easily. So, and a lot of times with these pumps, you're pumping something with the use of energy. So it's being pumped against its concentration gradient. So it's like going uphill. So that's why it needs a pump. It needs energy. So the first one is a calcium pump. And there's no definition needed here. It's just called a calcium pump. It requires ATP, all right? Because you're moving against the concentration gradient. But what I will want you to know is examples of where it's important in the human body. So it's important in all of your muscles, your heart and smooth muscle. So cardiac muscle. When someone is having a transplant and they're receiving a donor heart, obviously the donor's heart is not beating, okay? And they put the person on bypass, so all their fluids, their bodily fluids are on bypass. And then they put the donor heart in and they hook up all the plumbing. And then they stimulate it with a defibrillator to get the electrical activity going in the heart again. But if you don't have the heart bathe in a sodium and calcium solution of ions, it won't have the ions it needs to be stimulated to beat. The heart needs both sodium and calcium to beat, 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 beat. And if it doesn't have that, those ions are stimulatory. It causes the heart's pacemaker to start contracting. How many people knew you have to bathe the donor heart in an ion solution to get it? You can defibrillate it all day long, but if it doesn't have the ions it needs in the pacemaker to get it to start contracting, nothing will happen. So a calcium pump is pretty darn important in your muscles. It's important in your skeletal muscles too. All right. I'm not going to get into that here. I'll wait for the chapter on muscle physiology, but it's very important. Okay. The next pump is a hydrogen ion pump. It pumps hydrogen ions. Hydrogen ions are acidic. I think I mentioned it when I talked about pH in chapter 1. So here's an example. Important for parietal cells of the stomach to make acid. Okay, those cells pump hydrogen ions to make hydrochloric acid, HCl. So I don't know if you learned the parietal cells in anatomy, but they are your proton pumps in your stomach. They pump out a bunch of hydrogen ions to make hydrochloric acid, which is a pH of about 1 something, which is very powerful. Now, I've mentioned this before, the example, but in a different context. I said, if someone's having a heart attack and you run all these tests in EKG and it looks like they have not had a heart attack, you ask the patient, do you have gastroesophageal reflux disease or reflux? And they say, yeah, okay. That's when too much of these hydrogen ions are being pumped out, too much hydrochloric acid, or you have a weak cardiac sphincter between the esophagus and the stomach. And that acid backs up into the esophagus and you've got problems from that point on. So here's an example of a common prescribed, not some of them aren't prescribed anymore, treatment for acid reflux that's bad. All right. We're talking about people who have erosion of their esophagus lining. You got to stop that stuff, all right, before you have permanent damage. So Nexium, all right, the purple pill, basically slows down the parietal cells of the stomach, slows down hydrogen ion pumps. So you're not pumping out as much hydrochloric acid. So it's helping keep that acid down a little bit to protect their esophagus, all right? So that's an application of, here's a hydrogen ion pump. Here's where it's important and what it's doing. Here's a problem with this system, and here's a treatment for it. I'm going to do this a lot in physiology. I'm going to introduce a system to you, and you say, oh God, why do I have to know all this? Because when the system goes wrong, here's a pharmaceutical drug, and here's how it works. And if you know how the system works, you know how the drug works. And the reason I go over this with you is because you guys, as nurses in the future, are going to be spending more time with your patients, a huge amount of time compared to the doctor who comes in, looks at the, okay, we're going to try this. And they're really not that fast, but sometimes they are. You might spend as much as 15, 20 minutes talking with the nurse, all right? So when the doctor prescribes something, and I'm going off script here, but I'm trying to tell you why this stuff is important. They're going to say, well, that costs a lot of money. Why do I have to take that? Then one of the number one problems you're going to have in your future careers is patient compliance. They're not going to follow the directions and do it the right way. If you, as a nurse, can explain it to them, well, here's how this works. Here's what the problem is you have. Here's exactly how this drug works, and that's why it's going to work for you. You're going to have better patient compliance. So if you understand these systems and you can explain it in a simplified way to your patient, you're going to get better compliance because now they understand, right? Right. Okay. Way off script, but it's okay. Yes, we've got time. Okay. The last pump that I'm going to discuss is the sodium-potassium pump. And of course, the sodium symbol is Na and the potassium symbol is okay. All right. I forgot caladium. I forget the Latin that it comes from. Okay. So what happens is your cells pump 3 sodium out and 2 potassium into the cell. So here's the ratio. Sodium, the cells are be very careful. There's always a lot of sodium outside of the cell. When it comes into a cell, it's going to stimulate that cell. But when the cell is done being stimulated, it has to get its cell membrane potential back to resting. Okay, let me give you an example. Skeletal muscles. When they contract because the sodium channel is opened. And so that sodium floods into the cell like a big sodium tsunami. And that makes your muscles contract, your muscle cells. If you don't get that sodium out of the cell after the contraction happens, contraction keeps going. And now you've got seizures, all right? These ions have got to be kept in careful concentration inside the cells. So this is why this pump is very important to get yourself back to a resting position. Think of a skeletal muscle. It gets the chance to rest after it contracts. So you pump 3 sodium out of the cell, like the muscle cell, and bring in 2 potassium into the cell. Sodium is always stimulatory. Potassium makes cells kick back and relax. Okay, kick back. All right. You'll be very, very important for you later on. Okay. All cells use this to be able to rest. Okay. And so if you wanted to look at this, now cycling through, 3 sodium are being kicked out of the cell and 2 potassium will be coming in here. All right. But it takes energy. Okay. Are we good? We're almost at the end of this lecture. Told you it was a short one. Okay. Secondary active transport called coupled transport. Okay, the first example of this, it all takes ATP. We're still under active transport. Co-transport is known as symport because the passive transport of one ion across a membrane saves energy for the active transport of another substance across the membrane, but they're going in the same direction. They're either both entering the cell or they're both leaving the cell, usually entering. All right. So what we've got here, symport, the passive transport of sodium, because sodium is always in a high concentration outside your cell. Sodium is always beating down the door of your cell trying to get in. It doesn't take energy. But glucose, on the other hand, is a bigger molecule, and it's going to need some energy to fuel that to come in at the same time. So that's a good example. I'm going to give you a very good practical example in nursing for this. But before I do that, let's talk about antiport. Counter transport is called antiport because the passive transport of one ion saves energy for the active transport of another ion, but they're going in the opposite direction. So if sodium's coming in, something else is leaving. All right. They're just going like ships passing in the night. All right. But this one in particular, the symport or co-transport of sodium and glucose is very important in hydration. So let's say you've got a patient who's dehydrated, which can happen in the summertime, right? That patient comes in, they're dehydrated. You can tell very quickly, you can do some quick tests, say, yeah, you're dehydrated. Which do you think hydrates them better? A glass of pure water or a glass like Gatorade? The Gatorade, right? If you ever looked at the ingredients list of a Gatorade, they invented it in University of Florida to help hydrate their football players. Because scientifically, they found out that if you have some salt and some sugar in the liquid, it's going to hydrate the person better, faster. Okay. So let's say the patient come in and they're dehydrated and they're conscious. You don't stick an IV in them, right? Because first of all, it could introduce bacteria like MRSA, which you don't want to do it unless you have to. It's expensive to do that. If they're conscious, you want them to drink. Okay. But you want to give them something like oral rehydration salts, okay? Dissolve in 1 liter of drinking water, pure tap water. Look what's in it: sodium, potassium, and glucose. But this is important: sodium and glucose. It's going to hydrate them better, faster. So here's what's happening. Let's say someone, how could they be dehydrated if they weren't like working out in the sun? If you have chronic diarrhea, you're going to be losing a lot of fluids, especially children. Chronic diarrhea in an infant, they're going to be dehydrated very, very, very fast. Okay. Limits the ability of the intestines to reabsorb salt and water, leading to the risk of dehydration. I said life-threatening in a child, it is. But chronic diarrhea doesn't interfere with the co-transport, symport, of sodium and glucose in your intestines. So here's what happens in your intestines, your small intestines, all right, especially the duodenum, that's where glucose gets reabsorbed. So it's a tube that has a membrane around it, okay? If you consume solution that has both salt and glucose in it or sugar in it, what's going to happen is salt is going to cross the membrane first into your bloodstream, okay? Water by osmosis follows salt wherever it goes like a puppy. So if salt enters the bloodstream, water is going to follow that salt by osmosis, trying to dilute it. So by having salt move into your bloodstream, water from your intestines gets absorbed into your bloodstream. So now I've talked to you about solutes moving from high to low, simple diffusion, that's like salt. And now water moving, because wherever salt goes, water's going to follow like a puppy. That's how water moves in your body. Water doesn't move in your body usually until salt moves, and then it follows it. So that's how this symport of sodium and glucose hydrates a patient better than just pure water alone. It is passive diffusion and osmosis. Okay, so I think that's pretty relevant to nursing stuff, right? Yeah, now you know how it happens. Okay, so moving right along. Okay, bulk transport, bulk, think big things. It takes energy to move big things across the membrane because they're big. So all this requires energy. So we've got something called endocytosis, and that means moving large materials into a cell, large particles into a cell. Okay, so there's different types of endocytosis. Endo means within, and cyte means cell. The word literally means into a cell, endocytosis. So the first type of endocytosis, and I told you this is all introbio stuff. Oh, sorry. There's phagocytosis and pinocytosis. Well, phagocytosis, let me back up a little bit. Oh, I guess they're linked together. Okay. Phagocytosis is endocytosis of large particles. I'll put over here, I can put it here, how white blood cells consume a pathogen. That is something that's kind of an applied nursing concept. All of your white blood cells work by phagocytosis, which is the endocytosis of a large particle. So here we have a little bacterial cell, I think that's what it says it is, and here's a white blood cell, and it literally opens up its cell membrane and takes in that cell. And then it has enzymes that break down the cell and kill it. That's called phagocytosis. A phagocyte phage means to eat, and cell means cell. It's a cell that eats. A white blood cell is a phagocytic cell. It eats bacteria, viruses, things like that. It consumes it and breaks it down. That's why it's called phagocytosis. Okay, pinocytosis is the cell takes in fluid. So anyone, what's Pinot Grigio? It's a wine, right? And wine's a liquid. Pinocytosis, think Pinot Grigio, which is a wine, which is a liquid, and it's a cell taking in liquid. So that's how I remember that one. Now I know who drinks wine. Receptor-mediated endocytosis, I've got it written down. There's a receptor on the cell, and the cell is usually bound by a hormone or other molecule, causing the substance to enter the cell. Can you give me an example that we covered in chapter 1? And I even mentioned it again in lecture today, where a hormone binds to a cell membrane and it causes the receptor to open up and allow a large, relatively large particle. So I'm not talking about ions here. Glucose, okay? The hormone involved that binds to a cell receptor to allow glucose into your cells is insulin. This is how insulin works, okay? You've got receptors for insulin, and when they're bound, the cell opens up to allow glucose in. So that's a good example, all right? So I'll put here, example is insulin binds to a receptor to allow glucose into a cell. Even though glucose is a simple sugar, it's still regulated. It needs some, if you don't have the signal of insulin, the cells don't take in glucose. So this is one of those examples, perfect example of it. All right, how are we doing? Fantastic. I might only have one more slide, I don't know. But that covers all the vocab with that. Okay, and if I finish this PowerPoint today, I'll post the completed notes so you have all the notes. Okay, so here is, oh, there was one more, exocytosis. And let's see if I can move this. It won't. I know I wrote over it, and I'll try and move it when I complete my notes. I'll just shift this text up. Bulk movement of molecules out of a cell. Exo means exit. All right, so exocytosis literally means exiting the cell. So anytime your cells make a protein, like basically DNA is transcribed and then your ribosomes make a protein, and that protein is supposed to be shipped out of the cell, that's how it gets out. It's pretty big. Even if it's like an amino acid, it's still big. And so a vesicle takes it to the cell membrane, which invaginates and spits it out. So that's how our cells get products out of our cells. All right, so that ends most of the main vocab. Now here, and if I don't get to all of it today, obviously I'll wrap it around to Wednesday, but this is how all our cells function. Our cells, after they're stimulated, must rest. Think about skeletal muscles. If skeletal muscles can't rest after contracting, number one, your body temperature skyrockets and you get hyperthermia, and that can cook your brain. So that's not good, that's bad. So cells must be allowed to rest. So they must get down to a -70 millivolt potential. Notice how in this diagram, the sodium is much bigger than the sodium inside the cell. What they're trying to show you in this diagram is there's a lot of sodium. In fact, let me do it. A lot of sodium, lots. Very little inside your cell. Comparatively, your cell usually has more potassium ions inside. That's why they make the K big here. So if sodium is allowed into the cell, it will change the resting membrane to a stimulated membrane. It's called depolarization. The cell, whatever the cell does, it does it. If it's a muscle cell, it will contract. If it's a gland cell, it'll secrete something. It'll do something, all right? So inside of the cell has fixed number of negatively charged particles. That's what the negative things are, and some potassium. Sodium is always kept outside of the cell because if it's allowed in, it's going to cause an action potential. Skeletal muscles contract, cells secrete something. So the more, when a cell is stimulated, sodium will enter the cell. Potassium will leave the cell. Never mind. Thought it was a swamp fox message, but it isn't. So what happens is the inside of the cell goes from negative to positive for a brief moment in time, and it has an action potential. Muscles contract, cells secrete something. Once the cell has done that, you have to get the sodium out and bring potassium back in to make the cell more negative, and that's its resting potential. Okay? What kind of pump is going to be doing that? That one I just taught you. 3, after the cell's been stimulated to get it back to resting, 3 sodium are pumped out for every 2 potassium brought back in. And that will bring the cell membrane back to a negative resting state. Is that all? Have I explained that well enough from the pumps to this? Okay. All right. So here's what happens. I'm getting this concept here. So sodium is always stimulatory. But too much potassium is not good too. All right. So here's, and I've got a PDF. If you click this, when you're online looking at this PDF, you get to this clinical app, this place I stopped for today. And what it says, the resting cell potential is sensitive to changes in plasma potassium. If sodium is always stimulatory, potassium is always inhibitory. Potassium makes cells kick back and relax and make cells rest. Okay, if you have too much potassium in your bloodstream, it's called hyperkalemia. Hyper means high, emia means in the blood, and the K stands for potassium, the chemical symbol for potassium. When you have too much potassium in your bloodstream, cells stay rested. Your heart is very sensitive to too much potassium because potassium makes heart muscles rest. Your pacemaker rests. This can kill someone. Hyperkalemia can kill someone. In fact, patients who are on death row get an injection of potassium chloride solution, and it gives them hyperkalemia, and it makes their heart stop. So I'll finish this on Wednesday, but what I'm trying to impart on you, the last thing to think about is how much sodium, potassium chloride, and everything a cell allows in or out is going to have very profound effects on those cells. So these things are all important. All right, so we'll wrap this up. I think I got one slide after this for Wednesday. And then I believe we're starting on chapter 4. So make sure for Wednesday to bring that PowerPoint with you. And I'll see some of you in lab.